everyone. Let's study the various factors affecting enzyme activity. In the last video lecture, we have already studied that most of the enzymes are proteins. So now let's see what are the various conditions which will affect the rate of chemical reactions catalyzed by enzymes. So in short, all the factors which affect the enzyme activity. Now, the activity of an enzyme can be affected by change in the conditions which alter the tertiary structure of the protein. As proteins have different levels of structure, right? Primary, secondary, tertiary and quaternary. The tertiary structure is the shape of the protein, right? So, as enzymes are proteins, the enzyme will also have a characteristic shape, that is the tertiary level of protein structure. And factors like temperature, pH, right? If we change these conditions, then the shape of the protein can also change. And if the shape of the protein, that is in this case enzyme, changes, then substrate cannot bind to the enzyme and hence the rate of reaction will decrease. So, the various factors which affect the rate of reaction catalyzed by enzymes are temperature, pH, change in substrate concentration or binding of specific chemicals that regulate its activity. So, in this case, we will study chemicals called inhibitors which inhibit or slow down the rate of reaction. First, we will discuss temperature and pH. Let's understand how both these factors affect the rate of reaction or the enzyme activity. First, let's go through pH. So, if we take the whole range, like 0 to 40, then enzymes will act only in narrow range of pH. So, it will depend on the enzyme. Some enzymes will be active in acidic conditions, some will be active in basic conditions. It will totally depend on the specific enzyme. But enzymes always act in narrow range of pH and also temperature. Let's understand what is happening here in the graph. So, if you see, at low pH points, there is no activity of the enzyme. And at a particular pH point, the enzyme activity is started. And then slowly it goes on increasing as you increase the pH points, right? At a particular pH, let's say here, the activity is highest. The activity is highest. This particular pH point is called optimum pH. This is where the enzyme will do the best activity, highest activity. If you increase the pH further, then you can see the activity declines. And after a particular point, you will not have any activity like over here. So in this case, the enzyme is acting in this particular range. The minimum value where enzyme shows the activity, that particular pH will be minimum pH, and the maximum value where the enzyme shows activity will be the maximum pH. But in between the minimum and maximum pH value, you will have a particular pH value where the enzyme will have the highest activity, that is the peak in this graph. So this particular pH point is the optimum pH. Similar is the case of temperature. If we see at very low temperature, the enzyme is not showing any activity, right? At this particular temperature over here, the enzyme has started acting. And as you increase the temperature further, the activity is also increasing. As you can see, the graph is showing increase, right? And at a particular temperature, you can see there is highest activity, right? Activity will be highest. Again, this particular temperature point is called optimum temperature, where the enzyme will do the best activity. And as you increase the temperature further, what do we see? The activity declines, right? And it goes up to this particular temperature, okay? And after which you can see there is no activity. So again, for this particular enzyme, you will have some temperature, the lowest temperature where enzyme shows some activity, that is the minimum temperature. Then the highest temperature value where enzyme shows some activity, that is the maximum temperature. And in between minimum and maximum, you will have a particular temperature value where the enzyme will have highest activity. That temperature is optimum temperature. Enzyme will do the best activity at optimum pH and optimum temperature. Okay. So, what can we say regarding pH and temperature? Enzymes generally function in a narrow range of temperature and pH. Each enzyme shows its highest activity at a particular temperature and pH. And what do we call this temperature and pH? The optimum temperature and optimum pH, right? Activity declines both below and above the optimum value. Like in this case, you can see this is the optimum pH value, right? And activity is declining both below and above the optimum value. Same for the temperature. Now, specifically for temperature, at low temperatures, the enzymes will be preserved because as they are proteins, so at low temperature, the structure is preserved. But activity will be very low or like no activity, depends on how low the temperature is. But low temperature preserves the enzyme and high temperature destroys the enzymatic activity because proteins are denatured by heat, right? Denaturation will be because at high temperature, the hydrogen bonding which stabilizes the secondary and tertiary level of structure, this hydrogen bonding is disturbed. And if the hydrogen bonds are broken, then the final shape of the enzyme will not be maintained. And hence, the denaturation of enzyme and the enzyme activity decreases. Okay, this is how temperature and pH both affect the enzyme activity. Next important factor is concentration of substrate, right? Substrate is the compound on which enzymes act. So, enzymes bind to the substrate and convert the substrate into product. Let's understand how the concentration of substrate affects the enzyme activity. Okay? Look at this graph. On the x-axis, we have the substrate concentration. On the y-axis, we have the velocity of reaction depicted as V. Okay. Now, in this graph, what can we see? We start from this point over here, 0. Right. So, if there is 0 substrate, then there will be no product formation. So, the rate of reaction or the velocity will be also 0. As there is no substrate, so no product form and hence velocity 0. So, we start from this point. If you increase the substrate concentration, what can you see? There is increase in the rate of reaction. It's very obvious that if you add substrate, okay, in a tube which contains enzyme, then the enzyme will convert the substrate into product and slowly the amount of product will increase. So, more the substrate, more the product molecules form, right? So, the rate of reaction goes on increasing. But after a certain substrate concentration, you can see you increase the further concentration, there is no change in the velocity of enzyme activity, right? You can see that there is a stationary phase over here. That means, howsoever you increase the substrate concentration further, the velocity will not increase further. Now, why this is happening? This is because the enzymes, all the enzymes in the tube are saturated. Let's say in a tube, we have 100 enzyme molecules, right? First, you have, let's say, 10 substrate molecules. So, 10 enzymes will bind to the 10 substrate molecules and convert it into product. Let's say you add 20. No problem, as we have many enzymes, so now 20 enzymes will start acting at the same time and produce 20 products. 30, no problem, 30 substrate molecules can be acted upon simultaneously by 30 enzyme molecules, same for 50. So as you increase the substrate concentration, initially what we see, there is increase in the product formation, right? Per second, per minute, whatever the rate is, it is increasing. But what if you add now 100 molecules of substrate? As we have 100 enzymes and 100 substrates, what will happen? All will be converted into product. But what if now we add 110 molecules? If we add 110 molecules, but we have only 100 enzymes, so now whatever the rate is per minute, okay, whatever unit time you are taking, only 100 product molecules will form. Even if you add 200 substrate molecules, will the rate increase further? No. Only 100 product molecules will form, let's say per minute. Hence, what we can say that after a particular substrate concentration, the rate will not increase further and that maximum achievable velocity is Vmax, okay, as depicted here. This is the maximum achievable rate. So this is Vmax. Now on the y-axis, whatever will be the Vmax value, let's say in this case, let's take some hypothetical value 100. Then we can easily find its half on the y-axis. So it will be 50. We can write it as Vmax by 2, right? Half of Vmax. Simply interpolate it and what you can see here you will get a particular substrate concentration. Okay? This is constant and very specific value for each enzyme. It is variable, it varies from enzyme to enzyme, but it will be very specific for a particular enzyme. This particular value is called Km, right? Now, how can we define Km? Km, which is the Michaelis-Menten constant, okay, based on the scientist who explained this. Michaelis-Menten constant. It is the substrate concentration. As Km is on the x-axis and x-axis is the substrate concentration. So, Km is the substrate concentration when the rate of reaction is half of Vmax, right? That means 50% enzymes are bound to substrate. That particular substrate concentration will be Km. This is very important. So let's conclude what we have studied from the graph. With increase in substrate concentration, the velocity of the enzymatic reaction rises at first. So initially, when you start from zero, right? You start adding substrate, the rate is increasing. The reaction ultimately reaches a maximum velocity, that is Vmax, which is not exceeded by any further rise in the concentration of substrate. So, even if we add double the substrate, the rate will not increase further. This is because the enzyme molecules are fewer, and we have less enzymes and more substrate. So, enzymes are fewer than substrate molecule, and after saturation of these molecules, there are no free enzyme molecules to bind with the additional substrate molecule, and hence we reach this Vmax. If we take half of the Vmax value and simply draw this 90-degree angle, interpolate, then we get a particular substrate concentration that is constant for every enzyme, and that is known as Km, the Michaelis-Menten constant. Okay, so now the next factor which we will study is inhibitors. Okay, these are certain chemicals which when bound to enzyme will slow down or completely inhibit the rate of reaction. Okay, it will stop the reaction. The activity of an enzyme is also sensitive to the presence of specific chemicals that bind to the enzyme. These chemicals are nothing but inhibitors. See, when the binding of the chemical shuts off enzyme activity, the process is called inhibition and the chemical is called an inhibitor. So, we will study a particular type of inhibitor which we have for our NEET syllabus, that is competitive inhibitor. Now, we have studied that every enzyme has some specific shape and it has many pockets, right? So let's say this is an enzyme. A specific pocket where the substrate binds is the active site. So this is an enzyme. It has a pocket that is active site where the substrate binds. But now let's say we have another molecule which is very similar to the substrate, but it is not the substrate. This inhibitor molecule can easily bind to the active site and hence now the original substrate cannot bind, right? And as there is literally a competition between the inhibitor molecule and substrate molecule, like who will bind first to the active site. That is why such inhibitors are called competitive inhibitors. For any chemical to act as competitive inhibitor, it is very important that the structure of this competitive inhibitor and the substrate are very close. Okay, they are very similar. That is why only the inhibitor can easily bind to the active site and decrease the rate of reaction. So when the inhibitor closely resembles the substrate in its molecular structure and inhibits the activity of the enzyme, it is known as competitive inhibitor. We have classic example, that is inhibition of enzyme succinic dehydrogenase by malonate. Okay, there is an enzyme which is involved in Krebs cycle, succinic dehydrogenase. This enzyme is inhibited by malonate. So this is the competitive inhibitor of the enzyme. The actual substrate is succinic acid. Okay, but malonate is very similar to succinate structure, and hence malonate binds to the enzyme and acts as competitive inhibitor. Such competitive inhibitors are often used in the control of bacterial pathogens. Now imagine if we block a particular bacterial enzyme using inhibitor, then you can easily kill the bacteria or inhibit its growth. This is how competitive inhibitors are used to control the bacterial pathogens. So far we have studied the various factors which affect enzyme activity: temperature, pH, substrate concentration and inhibitors. Now it's time for knowledge drill. Try to answer the questions, okay, in comment section. First, fill in the blank. Temperature at which enzyme shows maximum activity is called dash. Okay, tell me in the comments. And next question, name the competitive inhibitor of enzyme succinate dehydrogenase. I hope you are clear with the various factors which affect enzyme activity. Thank you.